Hello everyone, welcome back to my YouTube channel. This is Fareen Khatun, and today we are going to discuss about the CRISPR-Cas9. And the CRISPR-Cas9 is play a very specific role in the field of molecular biology. It is a gene editing tool and performing very well. And it's not like limited, and I think most of the labs are using it, and if not, then they are going to plan to use it. So because it's giving very good results in the field of GMO production or transgenic plants. Okay, so in this video, we are going to learn about CRISPR-Cas9. So I'm going to share my screen now. Okay, wait a second. And I hope that it's all visible. Okay, let me check if everything is okay because I'm okay, now initiate. So we all know these are the very latest data about the CRISPR-Cas9 success, and I have recently studied it. It's like they modified a spider which produce a very like red fluorescent silk. Basically, they do some genome editing in the genome of spider, and now as they produce silk for their web making. Now that is in white in color, right? Now it's producing in the form of red fluorescent. So you can see in the darkness, which is the property of fluorescent, right? Now it's all moving towards cancer treatment. If it happens, then it's like very big wow, because they promise some gastrointestinal and some cancer therapy. It's in clinical trials, CRISPR showing so good results. And apart from that, for the environmental treatment also. So CRISPR is not limited for disease only. It's going beyond the imagination, and scientists are using and developing it. Now moving next. Okay. I hope that it's working. So what is CRISPR? CRISPR is clustered regularly interspaced short palindromic repeats. So CRISPR is not new thing. It's found in the bacteria earlier before in the form of defense, as we call adaptive immunity against fight against uh, some pathogen or some attack on the bacteria. It makes some it generates some response, and by using Cas9 protein. And now we use that Cas9 protein in the treatment, not treatment in as a form of genome editing. Okay. Now moving further, if we talk about history and milestone, like 1859, in this is like from the how it's genome editing working out, right? 1859, the Darwin origin of species comes, and then 1856, like Mendelian inheritance, when the genes come in the highlight, dominance and recessive. Then 1870, where nucleic acid, like going inside the DNA. In 1944, the genetic material, the debate between the RNA and the DNA, and DNA called the genetic material. Then 1953, the DNA structure. And then 1961 to 67, that was genetic code, which is amino acid sequence, right? And then 1970, like more deeper and deeper for restriction enzyme, Sanger sequencing, PCR, and then whole genome project. And the tools they are using like zinc finger, stylons, and now it's going to CRISPR's Cas9. So now moving towards, if I talk about the timeline, it's 1987, repeat discovered in E. coli, and 22, it's named CRISPR. 25, it's DNA match. 2012, it's uh, the Do- uh, Dodna, and the other one, Pioneer Editing. And 2020, it's won Nobel Prize. So how it works. So if I talk on the very, very basic level, how it's working out on, like, how it's DNA repair mechanism. It's like, no, we know that when there's a breakdown in the DNA sequence, there's the automatic repair mechanism initiate in the form of either non-homologous or homologous, right? We know that it's the very best, very, very basic. If it's non-homologous, then ideal mutation. If not, there's the repair template formed for the homologous sequence, and the repair goes on. Okay, so we are using that mechanism in the CRISPR. So its genome editing is for its zinc fingers, and talent it's a protein based, and if it for the CRISPR is the RNA based. So it's not a very big thing, but yeah, for the CRISPR-Cas9, you actually need the guide RNA. So I already told you, if we talk like how it working in the bacteria, it was the adaptive immunity. Here in this slide, it shows according to the how bacteriophage, if you know the bacteriophage, how it infect the bacteria. It release its genome, and that genome wants to overcome the host genome and control that host genome by making number of replication, and this is the how it survive in the host, or die out the host for its needs. So here the for counter that mechanism, uh, bacteria initiate a defense and call double strand breakdown for their survival mechanism, which is the key in the CRISPR-Cas9. So if we talk about, it's like I told you, the DNA repair is the main thing. There in this diagram you can see that how there is a palindromic sequence, and how guide RNA going there, attach CRISPR-Cas9, that big protein, and performing the mechanism and repair there. So when there is a repair, that the genome editing sequence think that it's their sequence, right? And the protein release inside the body. Okay, now moving further. CRISPR, the molecular, if what are the components are there? If I talk about what are the components are there, there is an array of identical repeats. Like there's a sequence which is have the palindromic sequence for recognize the CRISPR like Cas9 protein. Then DNA targeting spares where we are going to target, right? Operon is there, and the transitive RNA, which is the guide RNA. What are the components? For a Cas9 protein, there is adaptation, recognition of target site, two nucleus domains, which is RUVC, which is in a gray color, and NH, HNH, which is snake color, and that orange is showing the palindromic. So this is whole cluster is known as, not known as, it's showing as a Cas9 protein components. Okay, now there are components and cleavage. So in this diagram, it's showing type 1 and type 2, type 1, type 3, and the type 2 one. How it show cleavage. Okay, they make the complex. Oh, there is one thing, Cas9, and there is a complex form. How is the complex form, and they attach, and the breakdown happens. There is a complex happen on the palindromic sequence, and they cut. And now when they cut, there is a repair. There is a repair and DNA repair, and the repair goes for release of protein of our desire, because that repair with that sequence, which we want inside the our host, or where we want to make the transgenic plants. Oh, sorry, plant is my field, that's why I say plant, transgenic organism. So here are the palindromic sequence, a Cas9 protein C. Like for example, here is a S dot uh, phenogenes. For that, the palindromic sequence are NGG. So like that, where it is found that it will bind there. Okay, guide RNA work mechanism is there, cleavage is there, and then scissor works and cleavage happens, and there is a DNA repair mechanism, and the release of the protein happens. So this is the simply Cas9 or CRISPR works. Moving forward, we know the applications. Okay, I already told you how this is not limited in one field. It's in bacteria, fungi, animals, human cells, plants, and many more. For this diagram, I'm going to telling like it's a very very primitive like earlier research, not new, but for this by taking this as an example, I'm telling how it's in life. Like they choose for like study of disease genes. Here it's LRRK, like choose locus, then their guide RNA design or donut design, then transfection of the cells, and then the DSB generation for identification and characterization of mutant desired mutants. Like we get the bunch of, right, so we go test and we know who won the successfully mutant and who were not. We have, as a scientist, we have the characteristics. Okay, now moving forward, CRISPR-Cas9 has a genome editing, like because it's it working, right? Then regulation of gene expression, how it's regulating, and how we can see that the regulation, we have like imaging of genome loci, which is GFP, right? And now here, this one is a very, very recent example of successfully making a global genome edited rice variety. It's in India, IRI department, and the very first one is a Pusa rice DST1, which shows drought and salt tolerance. And moving forward, its second one is DRR Dhan 100 IIRR, which showed 90% yield increase, 20 days earlier maturity, and low fertilizer or drought पे भी अच्छा work करता है. Now, through this, we are moving towards precision agriculture, which means like what kind of land we have, we can grow the crop. We know the check the what we have, and we modify the crop according to that. So if there is a high humidity, okay, let's try to make a crop which survive under high humidity, and CRISPR going to help in that. So by doing such result in the field of research shows that we can do that. Okay, this is the future, and I hope that we are working on this very, very in great enhance and work for the world and not for the human community, not limited only a specific country. As a scientist, we are global. And these are the references. And I would like to say thank you very much for paying attention over here. And I'm going to stop now. And thanks for watching. Bye.